Hello, my name is Frank Chavez, and I'll be presenting a primary research article on the effects of N N-Dimethyltryptamine on rat behaviors relevant to depression and anxiety. Now, for some background, current antidepressants usually take a long time to start working, and they are particularly ineffective in about 30% of the population. N N-Dimethyltryptamine, also known as DMT, is one of the common compounds found in ayahuasca, which is a brew that has had huge historical and cultural claims of treatment. This study aims to find a new treatment that is fast-acting and also works well with patients that have treatment-resistant depression. It hopes to find results similar to ketamine and to also explore the specific mechanisms that might be producing these results. Although ayahuasca has shown promising results, there's more than just DMT in it. As you can see, there's also harmine and harmaline, which are monoamine oxidase inhibitors. Now, these compounds have been previously studied on their own and have shown pretty promising results just by themselves. However, this study aims to study specifically DMT, which is the main hallucinogenic compound in ayahuasca. This study will inject mice with DMT, wait about an hour for DMT to be completely processed out of the body, and then go through a series of tests such as Novelty induced locomotion, elevated plus mace, fear conditioning test, and also the forced swim test. The DMT that was used in the study was synthesized in the laboratory and administered to the mice via injection. These mice are 8 to 14 week old male sprig dolly rats. The first test that was done was to study novelty induced locomotion in these mice, in which they would put these mice in a box that looks much like this and study their movements for about 45 minutes, in which they would collect data on how much they were rearing, which is standing up, how long they were doing that for, the horizontal motions that they would make, as well as rotations. Researchers conducted this test by having mice injected with DMT and also having mice injected with the placebo. They would wait 1 hour and then put these mice into this chamber. The results indicated that mice treated with DMT had a decreased exploratory behavior while also traveling a much shorter distance and spent less time moving around than the control group. There was also no effect on thigmotaxis, which is poking the mice and seeing how their movement and orientation changes. The DMT-treated mice had a decrease in the amount of times they were rearing, as well as they spent less time doing so. They also had a decrease in the amount of abnormal repetitive behaviors, but not the total amount, which indicates that they spent less time in general doing these abnormal repetitive behaviors, but not the amount of times doing those behaviors. These results indicate that it produced a anxiogenic effect, which means that it actually increased anxiety, but it did not do so in the thigmotaxis measure. The next test that researchers used to assess anxiety is called the elevated plus mace. This is a beam that is elevated 50 centimeters above ground, and it has 2 open arms and 2 closed arms. The more time that these mice spend in the closed arms feeling protected indicates that they are more anxious. Once again, researchers got mice and treated them with DMT or a placebo, and then proceeded to wait 1 hour before placing them into this maze. These are a completely new set of mice, and these mice have not undergone any of the other tests. The results indicated that mice treated with DMT had less open arm entries and spent less time traveling in these open arm bridges. There was no difference between the amount of time that they spent traveling as well as the speed that they were traveling at, and there was also no difference in the amount of closed arm entries that they had. This suggests that there was no effect on the locomotion, but there was an effect causing more anxiety. For the fear conditioning test, mice received DMT or placebo on day 1, 1 hour before the trial. They then underwent auditory cues correlated with foot shocks for 3.5 minutes. On day 2, they underwent the same test except without any foot shocks. Then on day 3, mice were placed into a new environment and underwent the same test. The results for the fear conditioning test were that mice treated with DMT had no effect when compared to the placebo group on freezing prior to receiving the foot shocks. However, mice treated with DMT did spend more time freezing after receiving those foot shocks. There was also no effect on fear memory conditioning. For the fear-cued extinction test, mice underwent fear conditioning on day 1 and were given day 2 off. Then on day 3, mice were administered either DMT or placebo, and then placed into a new environment to undergo the same auditory cues. Then on day 4, these mice were placed back in the same environment with the same auditory cues, except no mice were administered any drug. It was on this day that data was collected on the percentage of time that mice spent freezing. The results for the cued fear extinction test were that mice treated with DMT had enhanced fear extinction. In other words, the tone caused mice treated with DMT to freeze a fewer amount of times. For the contextual fear extinction test, untreated mice were placed in an apparatus for 3 minutes prior to receiving a total of 6 foot shocks. They then had day 2 off. On day 3, mice were treated with either DMT or a placebo and placed back into the same environment for 10 minutes. This experiment was repeated on day 4 and 5. On day 6, the mice were then placed back again into the same environment for 10 minutes. It was on this day that data was collected based on the percentage of time mice spent freezing. The results of these tests show that there's no effect between the groups. However, previous ayahuasca research has showed positive results with these tests. This may be due to the other alkaloids that are present in ayahuasca, along with DMT. The final test that researchers conducted was the forced swim test, which on the first day, mice were placed in a cylindrical tube filled with water for 15 minutes. On the second day, they were placed back in that tube again, and they were recorded for 5 minutes. The data that was collected was the amount of times they spent climbing, the immobility or the swimming, the duration for these entire 5 minutes. Researchers again took mice and gave them either DMT or placebo. Except for this test, they also gave them ketamine as another marker of these results. What they did was they administered these drugs to these mice and waited 1 hour. Then they put the mice in the tube and recorded their movements for 5 minutes. What the results show is that DMT mice had an increased total time swimming and a decreased total time immobile. These results are consistent with the results produced from ketamine. There is also no difference in climbing between the group. The importance of this research is that there are now compounds that have a fast-acting antidepressant effect, as well as work well in patients with treatment-resistant depression. DMT is a molecule that has similar effects to ketamine. However, DMT works on the 5-HT receptors, unlike ketamine, which works on NMDA receptors. There is also evidence to suggest that DMT may cause neuroplasticity. This is shown by DMT still producing effects despite being completely cleared from the body. More research is still needed, but there are very promising results.